Hello, my name is Rob Hutchison and I'm the Chairman and CEO of Bioasis Technologies, Inc. Bioasis is a biotech company with an extensive patent portfolio surrounding a very unique physiological protein called P97 that is produced in the brains of patients who are suffering from Alzheimer's. Over the next number of months, we will confirm that P97, elevated levels of P97, is a biomarker and an indicator for physicians and doctors to be able to prescribe therapeutic drugs for those patients suffering from this debilitating disease. Additionally, Bioasis has recently confirmed that P97 can shuttle therapeutic drugs or large molecules across the blood-brain barrier and have them effectively delivered to the cellular level of the brain tenfold that of the drugs on their own. Bioasis has two scientific divisions. The first is the pursuit of the biomarker to help physicians in an aid to diagnose Alzheimer's. And the second division is the blood-brain barrier therapeutic delivery platform that will enable pharmaceutical companies to deliver their large molecule drugs in a more efficient and effective manner across the blood-brain barrier and into the cellular level of the brain. Bioasis was formed as a private corporation in October 2007, specifically to acquire from the University of British Columbia and the inventor, Professor Wilfred Jeffries, an extensive set of intellectual property containing a broad patent portfolio and over 20-plus years of both clinical and laboratory research. The intellectual property dates back to the early 1990s when Dr. Jeffries and his team at UBC discovered that there was a direct correlation to the concentration levels of a unique physiological protein called P97, with the scientific term melanin transferrin, and the dreadful disease Alzheimer's. In studies Dr. Jeffries performed at the University of British Columbia, they found that serum samples taken from persons whom had confirmed Alzheimer's post-mortem versus samples of physician-diagnosed Alzheimer patients, and finally versus the control samples, showed that the level of P97 increased with the advancement of the disease, with the highest levels in those post-mortem samples tested. With these early findings, Dr. Jeffries and his team embarked on running 3 clinical trials. The last one, published in 2001, was a fully double-blind study that was unblinded by an independent US-based laboratory. Subsequent to these 2 studies, 3 fully independent clinical trials were run: a 220-patient trial at the Department of Psychiatrics in Seoul, Korea, and a team from the University of Alabama. Both of these independent clinical trials confirmed the results of Dr. Jeffries, that P97 had a sensitivity and specificity of greater than 85%, and therefore may well be used as a biomarker or as a physician's aid to diagnose Alzheimer's. As I mentioned, the research behind P97 is some 20-plus years old, with an extensive laboratory research and numerous clinical trials conducted at both the University of British Columbia and by independent researchers, all confirming each other's results. So how large is the problem? Although numbers may vary, Alzheimer's is estimated to affect some 15 million people worldwide, and with the baby boomers entering 65 and plus, this number is expected to increase over the next 10 years dramatically. The cost associated with dementia in general is estimated to be over $300 billion annually US, with the overall cost to society being even higher. This is a huge problem, and it's only getting worse. Society and physicians need, 1, a reliable, quick aid to diagnose Alzheimer's, and 2, pharmaceutical companies need a companion diagnostic to measure the effectiveness of their therapeutics. Alzheimer's is not simply one disease. It is now recognized as a syndrome with a variety of causes and a variety of symptoms. The one stable constant is that a brain under attack by this disease or syndrome forms plaques on the cells of the brain, and the brain under attack produces P97 in greater concentration than a normal brain. Therein lies the correlation between the levels of P97 in blood, Alzheimer's, and the advancement of Alzheimer's. As you can see, the problem that faces society with regards to Alzheimer's is staggering. And with the baby boomers now surpassing age 65, the problem is only going to get worse. Doctors need a quick, safe, and reliable test for the early diagnosis of Alzheimer's so that they may commence therapeutic treatment as soon as possible and hopefully retard this disease. So what are the next steps? Bioasis's next steps have commenced with the engagement of the University of British Columbia and Dr. Jeffries' lab to develop an ELISA assay for commercialization, perform a confirmatory study that conforms to FDA requirements, and have our biostatistician then prepare and submit by a target date of June 2009 our FDA application to have P97 approved as a biomarker or as an aid to diagnose Alzheimer's. As part of the FDA process, Bioasis has engaged Anderson Forschung of Washington, D.C. to develop a cis-kappa assay based on proteomics to act as a certification assay for the commercial ELISA assay. Essentially, the FDA requires that a separate assay, other than the one you want to have certification on, confirms the result of the commercial assay. As you've just heard, Bioasis has a well-defined scientific and corporate plan to reach the FDA submission, and our capital resources to do so are adequate. Our other corporate objective deals with the therapeutic use of P97 to ferry drugs across the blood-brain barrier to the cellular level of the brain, or as we refer to it, as the blood-brain barrier delivery platform. Since early 2001, Dr. Jeffries and his team have been investigating the therapeutic aspects of P97 and have found that this physiological iron-binding protein has a unique capacity to shuttle therapeutic agents across the blood-brain barrier and deliver a payload of up to 10 times that of a drug not attached, or as the scientific community calls it, conjugated to P97. In simple terms, a drug conjugated with P97 is delivered through the blood-brain barrier to the cellular level of the brain at a concentration of up to 10 times that of a drug not conjugated with P97. It is widely accepted that a key to combating neurological diseases, such as inoperable brain cancer, is the concentration of the therapeutic agent that can be delivered to the diseased cells. So therefore, a tenfold increase is dramatic. There is a huge side benefit of P97 conjugated drugs. As I have mentioned, one of the major problems in fighting neurological disease is the amount of the drug that penetrates the blood-brain barrier. In many cases, it is a fraction of a percent. So what happens to the rest of the drugs? It settles in the major organs of the body, such as the heart, causing a condition known as cardiotoxicity. Our published peer-reviewed research found that P97 conjugated drugs did not settle in any concentration in the heart. Rather, it was expelled from the body. The therapeutic aspects of P97 were fully reported in our PLOS1 fully peer-reviewed manuscript published in June of 2008, of which a full text can be found on our website, bioasis.ca. The corporate plan for P97 as a therapeutic carrier is to engage and work with one or a number of pharmaceutical partners to pursue the research and clinical trials to fully develop P97 as a blood-brain barrier therapeutic delivery platform. As mentioned, Bioasis is not going to pursue this on their own, but rather find one or a number of biotech pharmaceutical partners to do so. An interesting aspect of this approach is that some potential pharmaceutical partners have indicated to us that a P97 conjugate with their therapeutic agent may in fact extend their drug's patent life, as our patent for P97 as a therapeutic carrier extends out to 2023. The opportunity for P97 as a therapeutic shuttle is immense. Drug companies have fought for years with this problem to be able to increase the effective dosage of their therapeutics to get to the cellular level to fight neurological diseases. P97 may have just solved that problem. I would like to now introduce our team to you. Dr. Wilfred Jeffries heads our scientific team and has been focused on the analysis of human-based proteins for over 25 years. He has vast publications and extensive peer-reviewed studies that speak for themselves. Dr. Terry Pearson, professor at the University of Victoria, has over 35 years' experience in biochemistry and is widely accepted as an expert in protein-based illnesses. Specifically, Dr. Pearson's and his research teams use proteins, microchemical techniques, and mass spectrometry as tools to identification of the molecular interactions. Dr. Lee Anderson, a member of our scientific board of advisors, was the chief science officer for a large-scale biology company trading on the NASDAQ stock exchange, whose proteomics division he founded in 1985. Prior to its merger in 1999 with Biosource Technologies, At LBS, he developed the first automated two-dimensional technology platform for proteomics research and pioneered a wide range of applications in drug discovery, toxicology, and surrogate markers. John McKay rounds out our science team and has vast experience within the pharmaceutical industry. John has taken a number of drugs from conception within the lab at GSK to full FDA approval and commercialization. John brings not only vast experience within the science community, but too as well has taken what is made in the lab to the commercial marketplace, a talent held by very few. David Clark, CA, is our CFO who has extensive public company experience and a strong financial background. Mr. George Saunders rounds out the board of directors. George sits on a number of public company boards and has a strong appreciation of corporate governance. And as CEO, I bring a balance between the scientist and the ultimate commercialization of their work. I'm the author of a number of patents and created and administered an aggressive patent portfolio at a company I founded called eCharge Corporation. While at eCharge, I held the positions of president and CEO and chief scientist, and as a part of the finance team, I assisted in raising over $90 million for that corporation. The key to Bioasis is to ensure that the right partners are engaged. That is my ultimate goal. Bioasis currently has sufficient funds to reach its 2009 both scientific and corporate plan. Currently, we have 18,300,000 shares issued and outstanding, and if all the warrants and options were exercised, that would increase to 29,300,000. In doing so, we would inject approximately $2.48 million into our corporate coffers. Our financial projections are based on a number of factors. 1, the completion of the ELISA assay, and 2, the FDA's submission by late June 2009. With these two factors in place, the company will commence an aggressive program to license initially a major U.S. laboratory to perform P97 tests. We are projecting that in late 2009, 2 such licenses will be entered into, leading us into a 2010 with a global licensing program. Additionally, the company intends to select a partner in 2009 to pursue the blood-brain barrier therapeutic delivery platform. Corporately, our objective is to license up to 15 international laboratories that will service our global clients. Thank you very much for allowing me to present to you Bioasis. If you need any further information, please go to our website, bioasis.ca, where all of the information can be found. Thank you.